Good morning, student. Today we are going to know about secondary growth in Bignonia stem and Dracena stem. So first of all, this Bignonia and Dracena showing a secondary structure that is not a normal growth in case of a climber or a perennial monocot. So first of all, monocotyledon can complete their life cycle in 3 months, 6 months or annually. But some exceptional cases like Dracena, they are perennial and they develop a one annual ring for each and every year. So first of all, on the stem of a Dracena, there is a circular swelling which is developed on each and every year. Whatever the leaves are present on that leaf nodal portion that can be fed or since down, fall down, and they left over their scars, that is a annual ring. So this is the Dracena plant, which is showy or cultivated plant. Likewise, this is a Bignonia plant. It is a woody climber and both of them are showing a anomalous secondary growth. So first of all, we are going to know what is meant by anomalous secondary growth. So in case of dicotyledon stem, in most of the cases have the normal structure in which ring of collateral vascular bundles are found. But at the time of secondary growth, inner and interfascicular cambium cut off xylem towards the inner side and phloem towards the outer side. So at that time, however, in a number of plants, abnormal conditions are met with abnormality is due to the two causes. First one, in some primary structure itself is abnormal. Means if a plant is in a primary developing condition at that time, if they develop a secondary features or secondary growth, that is a abnormal. While the primary structure is normal but secondary growth is abnormal. So such type or such causes or such type of region are also found in many more abnormal cases. The anomalous structure in dicot stem may be due to the presence of medullary or cortical bundles. Such condition is found in Borevia and are two rings of medullary bundles, inner of two, then rather big bundles in outer of generally 6 or more vascular bundles. Likewise, Borevia, one another plant is showing such type of conditions. So in case of Acheranthus, there are only two medullary bundles which in internodal part point their protoxylem towards the each other. Then Mirabilis, Bougainvillea and other members of the family Nyctanginaceae, medullary bundles are present. In Chenopodium, the medullary bundles remains embedded in parenchymatous pith. Here they are said to be leaf traces due to the presence of cortical bundles. So these are some reasons. Sometimes a ring of vascular bundle is found in cortical region. So cortical region is a cortical zone, and in that case there is within that zone there is a development of vascular bundle. This one is an abnormal condition. These bundles are maybe referred as cortical bundles or they may be interrupted as a leaf traces. The presence of such type of bundles has been studied in many families. like Bignoniaceae, Casuarinaceae, Cucurbitaceae, Proteaceae, Foliaceae and many others one. So here is the one example, that is a Bignonia. So what happened in this case? Due to the absence of vessels, the presence of vessels in angiosperm is a normal feature, but some genera like Balium, Dremes, Expormum, Vintera, Zygogynum, Trichoderma and Tetracedron do not possess vessels. Due to the abnormal behavior of normal cambium, sometimes when the normal cambium start cutting cells at several places in irregular fashion and form at some places much larger proportion of a xylem than phloem, and at other places more phloem than xylem, and a ring and furrowed cylinder is produced. They may be simple type, for example Bignoniaceae species, or a complex one as in Bignonia. So these are the some big examples of developing examples of a Bignonia. So due to the presence of phloem bundles, sometimes only phloem bundles are found developing between collateral bundles, for example Cuscuta, and sometimes due to the presence of scattered vascular bundles in dicot, for example Pepromola, Piper, Thalictrum, Nymphia, Papaver originals and Elastomensa. Due to presence to interxylary or included phloem in some plants like Combratum, Salvadora, Lipandenia, etc., the intercalary phloem occurs. Abnormal behavior of secondary cambium, which is due to the normal cambium with abnormal activity, for example Bignonia and Aristolochia. Sometimes abnormal cambium with abnormal activity, for example Amaranthus, Acheranthus, Borevia, Mirabilis and Chinopodium. So this is the diagram A, which shows young Bignonia stem. Then this is a B, which shows some more developing condition, that is a cambium, which is responsible for secondary growth. While in a C, the Bignonia stem showing formation of secondary xylem. So here is the cambium which going to be activate and developing a formation of secondary xylem and phloem. So these are the secondary xylem and this one is the secondary phloem, and this is the furthermore growth pattern of a Bignonia stem, that is a section D within medullary. Secondary phloem are developed over here. These are the xylem, and in this way again there is a development of different type of vascular bundles. So here is the cork, this is the phloem patches which are light in color, these are the darker patches, that is a xylem, and in between both of them there is a cambium ring which is alternate with each other in a height perspective. So this is the deep down cambium ring which is present in between phloem patch and xylem patch. Here is the complete xylem patch, that's why the patch is going to be a abnormal condition. So here is the original section or a microscopic photograph which shows the presence of a vascular bundle. Here is the presence of a sclerenchyma. These are the phloem. This is the cambium, and these are the xylums. And in between both of them, here is the development of light colored phloem. So this TS shows many ridges and furrows and reveals the following tissues from outside with inside. First of all, this Bignonia stem shows single layered epidermis which consists of rectangular cell. Then a thick cuticle is present at above the epidermal layers. A few multicellular hairs are also arising from some cells. It is well differentiated into collenchyma and parenchyma. Collenchyma is present below the epidermis in the ridges in young stem, but at maturity they are developed sclerenchyma. So these are the thick walled sclerenchyma, and these are the secondary phloem which are in a green color. Parenchyma is present below the sclerenchyma or collenchyma in the ridges and directly below the epidermis in the grooves. So here is the position of a sclerenchyma. In old stem, cortex consists of cork, cambium and cortex, while endodermis is undistinguishable from cortical cells. The cell lacks Casparian strips. So this is the characteristic of Bignonia. While in case of a pericycle of a Bignonia, it is sclerenchymatous patches. So these are the sclerenchyma patches which are nothing but a patch, and that's why there is a complete absence of a pericycle which is a discontinuous. In case of a vascular system, phloem, secondary phloem, xylem, primary xylem are present. 4 longitudinal furrows of secondary phloem are present which are wedged in between secondary xylem cylinder. So these are the 4 furrows ridges which are present at 4 different corners. Then vascular bundles are conjoined, collateral, open and endark. Primary phloem is crushed and present in small patches. So these are the primary phloem which are going to crush and present in a small patches, while secondary phloem is in the form of ring which remains intruded into the secondary xylem at 4 places. So here is the secondary phloem, this is the secondary phloem, this one again a secondary phloem, and this one is the peak. The Bignonia ugnis bars of sclerenchyma are present in the furrow of secondary phloem. So these are the furrows. Within this transverse section of a Bignonia stem, showing a diagrammatic representation of a Bignonia Annular secondary growth, which showing epidermis, parenchyma, sclerenchyma. These are the star sheath. This is the secondary phloem. This is the cambium ring. This is the secondary phloem. This one is again secondary phloem. These are the secondary xylem. These are the secondary xylem, and this one is the peak. So in this way, there is arrangement of Bignonia secondary growth, while cambium is a single layer present in between xylem and phloem and bent towards the inner side along the furrows of secondary phloem. So this diagram again showing a presence of ridges and furrows. Secondary xylem consists of vessel, tracheids, fibers and xylem parenchyma. These are the most important elements of a xylem, and due to the intrusion of the phloem at 4 places, secondary xylem is ridged and furrowed at 4 different places. Primary xylem is present Primary xylem is present close to the pith, facing its protoxylem towards the center. Its location is just opposite to the patches of primary phloem and the pith, which is thin-walled and parenchymatous. So this diagram showing the secondary growth, that is a phallogen. These are the lenticel. This is the epidermis. This one. Below the epidermis there is a presence of cork. Below that, in a cortical region, there is a presence of a fiber patches. These are the secondary phloem, and this is the cambium. These are the secondary xylem. This one is the vascular rays, and this is the central pith. Sometimes secondary growth, which can be formed due to the 4 furrows of the secondary phloem in the secondary xylem, is due to the abnormal functioning of cambium, which was behaving normally sometimes earlier. At 4 or more places, cambium produce less amount of secondary xylem towards inner side and large amount of secondary phloem towards the outside. Thus, 4 wedges of secondary phloem are formed. They intrude into the secondary xylem, and so xylem cylinder appears ridged and furrowed. Here is the some identification points, that is presence of vessel, that is the most important characteristic of angiosperm anatomy or internal structure, while multicellular epidermal hairs, conjoined collateral open and dark vascular bundles, which are the characteristic of stems. Vascular bundles are in ring, and presence of cambium, that is the characteristic of dicotyledon. Normal cambium with abnormal activity. Sometimes cambiums are the development of cambium is a normal condition, but that normal cambium can develop abnormal activity because of some many more reasons. So these are enlisted over here. The transverse section of young Bignonia stem is like that of typical dicot stem, but at that time secondary growth in the interfascicular cambium develop in the normal way, forming a complete ring of cambium. Now the cambium ring forms xylem towards inner side and phloem towards outside in the normal way. The amount of secondary xylem formed is much more as compared to the amount of secondary phloem formed. The cambium ring, after performing for a short time its normal function, which began certain point to give off internally only a very small quantity of xylem and external a large amount of phloem. As a result of this, deep wedge of irregularly bending phloem project into the outer xylem. In this way, normal cambium shows abnormality, as a result of which 4, 8 or more wedges are formed. The formation of cork cambium and cork take place in the normal way. Abnormal cambium with abnormal activity. In this case, normal cambium ceases its functioning after a very short period, and then an abnormal type of cambium development takes place from cortex or pericycle and behave in an abnormal way. For example, Amaranthus, Acheranthus, Borevia, Bougainvillea, Chinopodium. So in this way, there is a development of a cambium, and it may be a abnormal or anomalous. Here is the one another example which is belonging to the monocotyledon or monocots, that is the genus Dracena. In case of a Dracena stem, normally the vascular bundles of the monocotyledon stems are closed ones. Thus, due to the absence of cambium, they lack secondary growth in thickenings and the vascular system is wholly composed of primary tissues. But sometimes the bundles remains irregularly scattered in ground tissue, forming a actostele where the limits of cortex and other ground tissue can be hardly discerned. Some monocotyledons belonging to the family Liliaceae, mainly the arborescent ones like Dracena, Cordline, Agave, Alloy and other exhibit a peculiar type of secondary increase in thickness, and account of which is given here, that is in the regions which have ceased to elongate, some cells are occurring outside the vascular bundles which become meristematic and form the cambium. The secondary meristem originates in the cortex. In fact, deep layers of cortex or pericycle, through it is difficult to locate those zones in many stems. So here is the diagrammatic representation of a formation of a conjunctive tissue and leptocentric vascular bundle in a Dracena. So diagram A shows the cambium forms parenchyma only on the peripheral side and parenchyma and xylem on inner side alternately. So these are the parenchyma, this is the cambium and these circular xylems. Then in section B, the cambium situated above the xylem. So this is the xylem, and above that xylem there is a presence of a cambium. Below that there is a presence of a phloem, that is in a inward position. After a period of activity, the cambium situated above phloem in B form of a xylem. Thus, a leptocentric vascular bundle is formed. The parenchyma formed by the cambium on the inner side is conjunctive tissue. So, what happened in this case? Here is the parenchyma cell which are in a cross sign. These circular cells are xylem, and this long elongated ring-like strip is nothing but an cambium. So over here is the presence of a parenchyma. Below that parenchyma, here is the presence of a cambium, and these are the xylems. In between cambium and xylem there is a small patch of a phloem, and here is the parenchyma. This a small hemisphere again developed into a ring-like structure. So this is the leptocentric vascular bundle. Within at a center there is a presence of a phloem, and phloems get surrounded by the xylem. These are the conjunctive tissues, these are the parenchyma, and this is the ring-like cambium. This such type of arrangement is called as a leptocentric vascular bundle. So the cambium cells are fusiform, rectangular in shape. Instead of forming phloem and xylem on the outer and inner side as in normal condition, but the cambium cells go on dividing and producing secondary tissue on the inner side first, and later small amount of new tissues are cut off on the outer side as well. That is a condition which is rather than normal condition. So those formed on the inner side differentiated into oval shaped vascular bundles and radially arranged parenchyma cells. These parenchyma cells in which the vascular bundle remain embedded are said to be constitute the conjunctive tissues. The radial arrangement of the parenchyma cell of a conjunctive tissue is due to their origin by tangential division of the cambial cell. What is meant by tangential division? That is a transfer division. So they may be easily distinguished from the irregularly arranged parenchyma of the primary ground tissue. They may be thin walled or thick walled. So at that time, few derivatives of the cambium divide longitudinally, initially anticlinally, then periclinally, and then horizontally to form xylem and phloem elements of the secondary vascular bundle. These differ from the primary ones in presence of small amount of phloem and in absence of annular and spiral protoxylem elements. The secondary bundles are mostly amphivalve. Some of them are bicollateral as well. The small amount of phloem consists of short sieve tubes, companion cell, and parenchyma. The xylem is made of only tracheids, usually with the scalariform thickenings, and small amount of xylem parenchyma which have lignified walls. The tissues cut off by the cambium cell on the outer side are scanty in amount and are parenchymatous in nature. The primary bundles are comparatively large. They are also mostly amphivalve or rather rarely collateral ones. In aloe vera, coconut, and royal palm, peridium is formed in the same manner as in the dicotyledons, but a special type of protective tissue is produced in the majority of monocotyledons with secondary increase in thickness. So this is the actual transverse section of a Bignonia Dracaena stem. In this case, parenchyma cells in the deeper layers of the cortex divide periclinally several times and produce radially arranged cells, suggesting something like a storied meristem. The series of cells thus formed undergo suberization and differentiated into a cork cell. These tissues are called as storied cork mainly because of its storied appearance. So this is the transverse section of a Dracena. This is the epidermis. This one is the cortex, and within that cortex or near to that cortex, here is the many more vascular bundles which are irregularly scattered in a cortical region. The apical meristem or shoot apex produce only a part of the primary body, a central column of a parenchyma and vascular strands. Another meristem occurs just beneath the leaf primordia, forming a mantle-like tissue region. This is known as a primary thickening meristem. So, due to that meristem, the cells of the same divide periclinally and forms rows of the cell which differentiate into the ground parenchyma transferred by the procambium strands. The latter ultimately develop into the vascular bundle. There are further enlargement and divisions of the ground parenchyma causing increase in the thickness. So in this way there is a development of vascular bundles, and thus the primary thickening meristem produce the main bulk of stem tissues, that is ground parenchyma and most of the vascular bundles. The thickening is due to the activity of two meristems stated above. So this is the transverse section, diagrammatic representation of a Dracena stem. This one is the epidermis, this is the cork cell, this one is the cortex, this is the cambium ring, these are the conjunctive tissues, this one is the secondary vascular bundle, These are the ground tissue, which is a primary in origin. These are the vascular bundle, which are primary vascular bundle. Secondary vascular bundle are always present at the periphery, while primary vascular bundles are present always toward the center. These are the phloem, which are present at the center, and around them there is a presence of a xylem. Secondary thickenings with the meristem occur in number of monocotyledon species such as Xanthoria, Dracena, Codline, Aloe vera, Yucca, Knigna, Dioscorea, etc. This meristem is a type of vascular cambium which originates in the parenchyma cell present on the peripheral side of the entire mass of a primary vascular bundle. This region may be the inner layer of cortex or pericycle. This lateral meristem is distant from the same stem apex in seedling stage. It develops in continuity to the primary thickening meristem. In mature plant, these two meristems are found to be discontinuous. This is the enlarged diagram of a vascular bundle or a complete Dracena stem. So here is the vascular bundle. This is the vascular elements, that is xylem. These are the phloem. This one is the sclerenchyma sheath, which is also called as a bundle sheath, and this is the bundle, means a vascular bundle. So here is the darker patches is nothing but an xylem. Here is the lighter patches, these are the phloem, and xylem and phloem are surrounded by the bundle sheath cells. This is the complete diagrammatic representation, which also again shows epidermis, cuticle. These are the cork. This one is the cork cambium. This is the cortex. These are the secretory cells. These are the secondary parenchyma. This is the cambium, which is responsible for the growth of vascular bundle. This cork cambium is responsible for the development of a perider. So in this way, Dracena showing anomalous secondary thickenings. So one of the outer layers of the cell from the ground tissue become meristematic and function as a cambium. The cambium found in the region which has cis elongating, and the activity of this cambium is more on the inner side and very little on the outer side. It forms only parenchyma. On the inner side it forms xylem and parenchyma in alternate patches. The inner parenchymatous cells are called as a conjunctive tissues. After a short while, the activity of a cambium on inner side changes and above the xylem it start forming phloem and then again xylem. The xylem formed earlier has bigger vessels. In this way, around each vascular bundle is developed sclerenchymatous sheath, which is also called as a bundle sheath. The cambium after some times alter its activity and forms xylem on the inner side at those places where it was previously forming the parenchyma, and parenchyma in place of a xylem. Similar to the earlier cases, again by change in the activity it forms a ring of vascular bundle. So in this way, alternately they can form their secondary growth. So activity of cambium goes on changing regularly and more rings of vascular bundles are formed. Cork cambium is formed below the hypodermis and forms cork, and cork cambium is normal function. So this is the Dracena plants, which are different type of varieties, because this plant is a perennial monocotyledon showy plant or cultivated plant, ornamental plant which is house plant, and this is the Bignonia, which is a ornamental garden plant. So both of them showing a abnormal secondary thickenings or abnormal secondary growth. That's why it is called as a anomalies. So thank you for watching my lecture.